Hello, everyone. Welcome to our lecture today. Today, we're going to dive into one of the most elegant and crucial concepts in biochemistry, allosteric enzymes. While we've talked about enzymes before, today we'll see how the cell has a sophisticated remote control system for these molecular machines. This isn't just about how a substrate fits into an active site. It's about how entire metabolic pathways are finely tuned and regulated. For our biology students, this is fundamental to understanding cellular metabolism, and for our pharmacy students, it's a critical concept for how many modern drugs are designed and function. Let's get started. Let's start with a brief review to get everyone on the same page. As you're well aware from your prior courses, enzymes are biological catalysts, which means they dramatically increase the rate of biochemical reactions without being consumed in the process. They're typically large, complex protein molecules with a very specific three-dimensional shape. This shape is what gives them their remarkable specificity, allowing them to bind to a particular molecule, the substrate, at a special pocket on their surface called the active site. The modern understanding of this interaction is the induced fit model, which suggests that the active site isn't a rigid mold, but rather a flexible structure that changes its conformation slightly to perfectly cradle the substrate upon binding. This conformational change is what facilitates the chemical reaction. But here's the critical question. If an enzyme is so good at its job, and its job is to speed up a reaction, what prevents the cell from constantly producing a product it no longer needs? How does a cell ensure it's not wasting valuable energy and resources? The answer lies in sophisticated regulatory mechanisms that act as the cell's master control system, a system that goes beyond just blocking the active site itself. The need for enzyme regulation is truly at the heart of cellular survival and metabolic control. A cell's metabolism is an incredibly complex network of interconnected pathways. Imagine a city's traffic flow. You can't have every road open and every car driving at top speed all the time. You need traffic lights, speed limits, and on-off ramps to manage the flow and prevent gridlock. Similarly, a cell must be able to modulate its metabolic traffic to maintain a state of homeostasis, a stable internal equilibrium. This prevents the wasteful overproduction of products that are already in high supply and allows the cell to respond rapidly to changing environmental conditions or internal demands. We've previously discussed simple forms of regulation, such as competitive inhibition, where an inhibitor molecule directly competes with the substrate for the active site, or non-competitive inhibition, where an inhibitor binds elsewhere but still physically blocks the active site. These are effective, but they are often blunt instruments. Today, our focus is on a much more subtle and often more powerful form of control, allosteric regulation. This mechanism allows for a nuanced, highly dynamic, and often cooperative level of control that competitive inhibition simply cannot provide. It's a game changer in how we think about cellular feedback loops. So, what precisely distinguishes an allosteric enzyme from a simple one? The name itself gives us the biggest clue. Allosteric comes from the Greek roots allos, meaning other, and stereos, meaning site or shape. Put together, it describes an enzyme that possesses a regulatory site that is distinct and physically separate from its active site. This is a critical distinction. This special regulatory location is known as the allosteric site or regulatory site. When a specific molecule, which we call an allosteric modulator or effector, binds to this other site, it triggers a conformational change in the enzyme. Think of it not as a direct blockage but as a remote control effect. The binding at one location causes a structural shift that reverberates through the entire protein. This ripple effect changes the shape of the active site, either making it more or less receptive to its substrate. It's a brilliant design because the active and regulatory sites don't have to be physically close, yet a molecule binding at the allosteric site can have a profound impact on the enzyme's catalytic efficiency. It's a very clever way for the cell to transmit a signal across a protein molecule. These allosteric modulators, or effectors, are the signals that tell the enzyme what to do. They fall into two main categories, defined by their action. First, we have allosteric activators. When an activator binds to the allosteric site, it stabilizes the enzyme in its high affinity, or active state. This conformational change makes the active site a better, more perfect fit for the substrate, thereby increasing the enzyme's catalytic activity. You can think of it as a signal to open for business or to increase the speed limit. On the other side, we have allosteric inhibitors. When an inhibitor binds to the allosteric site, it stabilizes the enzyme in its low affinity, or inactive state. This structural shift makes the active site less receptive to the substrate, effectively decreasing the enzyme's activity. The inhibitor sends a message to slow down or shut down. It's important to note how this differs from competitive inhibition. A competitive inhibitor literally blocks the active site. An allosteric inhibitor simply changes the active site's shape so that the substrate can no longer bind efficiently. This nuance is why allosteric regulation is such a powerful tool in cellular metabolism. To truly grasp how allosteric regulation works, we need to think about the enzyme structure in a more dynamic way. Instead of a single, static shape, allosteric enzymes are best described as existing in a rapid, reversible equilibrium between at least two distinct conformations. We refer to these as the tense state, or T state, and the relaxed state, or R state. The T state is the low-affinity conformation. Its active site is in a shape that is not well suited for substrate binding, so the enzyme is essentially turned off. The R state is the high-affinity conformation. Its active site is perfectly shaped to bind the substrate, making the enzyme turned on. In the absence of a modulator, the enzyme fluctuates between these two states. The allosteric activators and inhibitors we just discussed work by shifting this fundamental equilibrium. An activator binds and locks the enzyme in its R state, tipping the balance heavily toward the active form. Conversely, an inhibitor binds and locks the enzyme in its T state, pushing the equilibrium toward the inactive form. This elegant, reversible, and dynamic model explains how a cell can finely tune the activity of an enzyme in real time. Let's talk about the physical architecture of these enzymes, because their structure is directly responsible for their function. A key feature is that most allosteric enzymes are oligomeric, which means they are made up of multiple protein subunits, or polypeptide chains. This multi-subunit structure is not a coincidence. It's essential for their regulatory function. Each subunit can contain an active site and, in many cases, its own allosteric site. The subunits are held together by non-covalent interactions, but they are not independent entities. The binding of a modulator or a substrate to one subunit can physically and structurally influence the conformation of the other subunits. This inter-subunit communication is often referred to as allosteric communication and is a fundamental aspect of their mechanism. It's this intricate physical connection between the subunits that allows for a phenomenon called cooperative binding. Without this oligomeric structure, the powerful cooperative effects we're about to discuss would simply not be possible. Cooperative binding is arguably the most fascinating feature of allosteric enzymes, and it's what gives them their unique kinetic profile. Here's the concept. The binding of one substrate molecule to a single subunit of the enzyme increases the binding affinity of the other subunits for the substrate. It's a chain reaction of activation. Imagine the first substrate molecule binding to the first subunit. This binding event causes a conformational change that is communicated to the other subunits, making their active sites more receptive to the substrate. It's like a domino effect or a series of interlocking keys, where the first key you turn makes the others easier to turn. This is an incredibly powerful biological tool.
Why?
Because it means the enzyme's response to substrate concentration is not a gradual curve, but a rapid, almost all-or-nothing switch. A small increase in substrate concentration can trigger a dramatic increase in enzyme activity. This allows the cell to turn on a metabolic pathway very quickly once a certain threshold of substrate has been reached, making the regulation process extremely sensitive and efficient. The unique behavior of allosteric enzymes is visually represented in their kinetic plots, which look very different from the ones we're used to seeing. For a typical enzyme that follows Michaelis-Menten kinetics, the plot of reaction velocity versus substrate concentration is a classic hyperbolic curve. This curve rises steadily and then plateaus as the enzyme becomes saturated with the substrate. It's a simple, predictable relationship. However, for an allosteric enzyme that exhibits cooperative binding, the kinetic plot is sigmoidal or S-shaped. The curve starts with a slow initial rate, but once the substrate concentration reaches a critical point, the rate increases very steeply before leveling off at its maximum velocity. This S-shape is the kinetic signature of cooperativity. It visually demonstrates the switch-like behavior we just discussed. A small change in substrate concentration around the inflection point of the curve results in a large change in reaction rate, providing the cell with a highly sensitive on-off mechanism that is perfect for regulating key metabolic checkpoints. Now let's see how allosteric modulators influence this kinetic plot, because this is where we see the power of allosteric regulation in action. An allosteric activator binds and shifts the sigmoidal curve to the left. What does this mean biochemically? It means that a lower concentration of substrate is now needed to achieve the same reaction velocity. The activator has essentially increased the enzyme's apparent affinity for the substrate, effectively decreasing its apparent Km. It makes the enzyme more efficient, even at lower substrate concentrations. Conversely, an allosteric inhibitor binds and shifts the sigmoidal curve to the right. This signifies that a higher concentration of substrate is now required to achieve the same reaction velocity. The inhibitor has decreased the enzyme's apparent affinity for the substrate, increasing its apparent Km. These shifts beautifully illustrate how the cell can precisely and reversibly tune the speed of a reaction simply by regulating the levels of these allosteric modulators. This graphical representation is an invaluable tool for both biochemists and pharmacologists. One of the most powerful and widely used applications of allosteric regulation in living organisms is a concept called feedback inhibition. This is a classic example of a negative feedback loop that maintains cellular homeostasis. Here's how it works. The end product of a long metabolic pathway acts as an allosteric inhibitor for the very first enzyme in that same pathway. Think of a thermostat in your home. When the temperature gets too high, the product is in abundance. The thermostat sends a signal to the furnace to turn off. The end product inhibits the first enzyme. This prevents the system from overheating. Similarly, when the cell has synthesized enough of a particular amino acid or nucleotide, the excess product can bind to and inhibit the first committed enzyme in its own synthesis pathway. This is a brilliant and economical strategy. It prevents the cell from wasting precious energy and starting materials on producing something it already has in abundance. It's an elegant form of self-regulation that's found throughout all forms of life. Let's put this concept into a concrete biological example. Consider the metabolic pathway for synthesizing the essential amino acid isoleucine, starting from another amino acid, threonine. The first enzyme in this multi-step pathway is called threonine deaminase. This enzyme catalyzes the first irreversible or committed step in the pathway, the point of no return. Therefore, it is the perfect control point for the entire pathway. Now, the final product of this series of reactions is isoleucine. As the cell synthesizes more and more isoleucine, its concentration increases. When the isoleucine levels reach a certain threshold, the isoleucine molecules themselves act as a powerful allosteric inhibitor. They bind to the allosteric site of threonine deaminase, causing a conformational change that shuts the enzyme down. This immediately halts the entire synthesis pathway. Once the cell uses up its supply of isoleucine and the concentration drops, the inhibition is released, and the pathway can begin again. This is a perfect, textbook example of feedback inhibition maintaining metabolic balance. So, why is all this so important for those of you in pharmacy? Well, allosteric sites have emerged as incredibly important drug targets, often preferred over active sites. There are several key reasons for this. First, allosteric sites are often less conserved across different species than active sites. This means a drug can be designed to be a highly selective allosteric inhibitor for a specific bacterial or viral enzyme, for instance, without affecting its human counterpart. This specificity leads to fewer off-target effects and, therefore, fewer side effects. Second, allosteric modulation can be more subtle than active site inhibition. Instead of completely turning an enzyme off, an allosteric modulator can simply change its kinetic properties, for example, by shifting the sigmoidal curve to the right. This fine-tuning can be very useful. We're seeing this in the development of new anti-cancer drugs, for example, which target allosteric sites on key enzymes to slow down cancer cell proliferation without completely shutting down vital cellular processes throughout the body. The future of medicine is increasingly focused on this nuanced and highly specific form of targeting. To wrap up our discussion, let's consolidate the key points. Allosteric enzymes are distinct because they possess a separate regulatory site. They exist in a dynamic equilibrium between two conformations, the tense state, low affinity, and the relaxed state, high affinity. Allosteric modulators, or effectors, bind to the allosteric site and shift this equilibrium, either activating or inhibiting the enzyme. Due to their multi-subunit structure, they often exhibit cooperative binding, leading to their characteristic sigmoidal kinetic curve. Finally, this sophisticated system allows for crucial biological functions like feedback inhibition, where the end product of a pathway inhibits the first enzyme, ensuring the cell's metabolic processes are both efficient and well-regulated. This is far more than just a molecular mechanism. It's a fundamental principle of how life maintains order and responds to its environment. I hope this lecture has illuminated for you the elegance and efficiency of allosteric regulation. Thank you all for your attention. Now, I'd be happy to answer any questions you might have.